Chapter 6: Communication, Integration, and Homeostasis. This chapter highlights 5 essential topics related to physiological communication and control mechanisms. Cell-to-cell communication. How cells communicate to maintain homeostasis, with signals traveling across short or long distances using chemical, electrical, or mechanical means. Signal pathways, the core components of signaling processes, from receptor activation to cellular responses. These pathways involve ligand binding and intracellular messengers. Novel signal molecules, unconventional molecules like calcium ions and gases, including nitric oxide, NO, that influence cellular processes and responses. Modulation of signal pathways, understanding how cells adjust sensitivity to signals through receptor regulation, competition, and the actions of agonists and antagonists. Homeostatic reflex pathways, complex control loops integrating the nervous and endocrine systems, which regulate variables like temperature, blood pressure, and glucose levels. This foundation allows for an integrated understanding of physiological responses and the intricate communication between systems. To maintain balance in the body, or homeostasis, cells communicate using 2 major types of signals: electrical and chemical. Electrical signals rely on changes in a cell's membrane potential, essentially, the difference in charge inside and outside the cell. These changes are key for rapid communication, especially in the nervous system. But most communication in the body occurs through chemical signals, like hormones. These signals are secreted by cells into the extracellular fluid, or ECF, and then travel to reach their target cells. Once there, they act as ligands, special molecules that bind to receptors on or inside the target cell to trigger specific responses. Now, target cells don't respond to just any signal. They have specific receptors tuned to recognize particular ligands. This specificity is critical, but it's only one part of how chemical signals work. When ligands bind to their receptors, they follow important rules: specificity, affinity, competition, and saturation. Specificity ensures the signal binds to the right target. Affinity measures how strongly they bind. Competition can occur when multiple ligands vie for the same receptor, and saturation happens when all available receptors are occupied, limiting the response. Together, these mechanisms allow cell-to-cell communication to be efficient and precise, ensuring that the body's complex processes stay in balance. When we think about how cells communicate, it's important to distinguish between local communication and long-distance communication. Let's start with local communication. Cells that are close to one another have 3 primary ways to send signals. Gap junctions form direct connections between the cytoplasm of neighboring cells. These tiny bridges allow ions and small molecules to pass freely between cells, enabling almost instant communication. Contact-dependent signals require the cells to be in direct physical contact. The signaling molecules remain bound to the membrane of one cell and bind directly to receptors on the target cell when they touch. Chemical signals can diffuse through the extracellular fluid to reach nearby cells. These signals don't require direct contact and allow cells to interact even when they're not physically touching. Now, when signals need to travel longer distances, the body relies on both chemical and electrical signals. Hormones, which are chemical messengers, travel through the bloodstream to reach distant target cells, while electrical signals transmitted by nerve cells can rapidly span long distances to coordinate activities. This combination of local and long-distance communication ensures that cells can work together to respond efficiently to the body's changing needs. In this figure, we see 3 key mechanisms of local cell communication: gap junctions, contact-dependent signaling, and paracrine/autocrine signaling. In panel A, we have gap junctions. These structures form direct cytoplasmic connections between adjacent cells. Ions and small molecules pass freely through these connections, allowing cells to synchronize their activities. This is particularly common in tissues like cardiac muscle, where rapid communication is essential for coordinated contractions. In panel B, we see contact-dependent signals. Here, Membrane-bound signaling molecules on one cell interact directly with receptors on a neighboring cell. This type of signaling is critical in immune responses and cell development, where specific cell-to-cell contact triggers precise outcomes. In Panel C, autocrine and paracrine signaling demonstrate how chemical signals can work locally without direct contact. In autocrine signaling, a cell releases a signal that acts on itself, essentially sending feedback to regulate its own activity. In paracrine signaling, the chemical signal diffuses to neighboring cells, affecting their function. Think of growth factors, which are released locally to stimulate cell growth and repair. Together, these local communication strategies allow cells to coordinate their actions efficiently, whether they're responding to immediate environmental changes or regulating longer-term processes. Long-distance communication within the body relies on both chemical and electrical signals to ensure messages reach their intended targets, whether near or far. Let's start with chemical communication. Hormones, secreted by endocrine cells, are a prime example of long-distance chemical messengers. These hormones travel through the bloodstream to influence distant target cells. In addition to traditional hormones, we also have neurohormones, hormones released by neurons, such as oxytocin or vasopressin, which combine the roles of the nervous and endocrine systems. Now, electrical communication is predominantly handled by the nervous system, which integrates both electrical and chemical mechanisms. Electrical signals, known as action potentials, rapidly travel down nerve fibers, acting like an electrical impulse that can span long distances. When these signals reach the end of a neuron, they trigger the release of neurotransmitters, the chemical messengers that transmit signals between neurons or to target cells like muscles or glands. This combination of action potentials for rapid transmission and neurotransmitters for targeted signaling ensures that the body's responses can be both fast and specific. Whether it's a reflexive muscle contraction or a hormone-driven metabolic adjustment, long-distance communication is vital to maintaining proper function and homeostasis. This figure highlights the key mechanisms involved in long-distance communication, showing how signals can travel through the body using both the endocrine and nervous systems. On the left for D, we see the endocrine system in action. Endocrine cells secrete hormones directly into the bloodstream. These hormones circulate throughout the body, but only trigger a response in cells that have the appropriate receptors. For example, cells without a receptor for a specific hormone will ignore the signal, while those with a receptor will bind to the hormone and initiate the desired response. This selective process ensures precise control over long-distance signaling. In the middle for, we see the role of neurotransmitters in the nervous system. Here, an electrical signal travels down the neuron, and upon reaching the synapse, triggers the release of neurotransmitters. These chemicals diffuse across the small gap to the target cell, binding to receptors and generating a specific response, such as muscle contraction or glandular secretion. On the right for F, we see the action of neurohormones, a hybrid mechanism involving both the nervous and endocrine systems. In this case, neurons release chemicals directly into the bloodstream, where they travel to distant target cells. Just like traditional hormones, neurohormones only affect cells with the correct receptors, ensuring their signals are targeted and effective. These coordinated mechanisms demonstrate how the body integrates electrical and chemical signals to maintain communication across vast distances, keeping organs and systems functioning harmoniously. Cytokines are critical signaling molecules in the body, acting as versatile messengers that can function both locally and over long distances. These small peptides are synthesized and secreted by virtually all nucleated cells in response to specific stimuli. Examples of cytokines include interferons, interleukins, colony-stimulating factors, growth factors, tumor necrosis factors, and chemokines. Each plays a specialized role in maintaining cellular and systemic balance. During development and differentiation, cytokines typically act as autocrine or paracrine signals. For instance, in the bone marrow, they help regulate processes such as the production and maturation of blood cells. However, cytokines are perhaps best known for their role in the immune response. When the body encounters an infection or injury, cytokines mediate inflammation by coordinating the activity of immune cells. They recruit white blood cells, enhance the production of antibodies, and trigger a cascade of defense mechanisms. This dual functionality, acting both locally and at a distance, makes cytokines vital to ensuring the body responds appropriately to various stimuli, maintaining overall homeostasis while addressing specific challenges like injury, infection, or cellular growth. Let's talk about signal pathways, the process through which cells communicate and respond to chemical signals, whether locally or across long distances. For a cell to respond to a signal, it must have a specific receptor that recognizes the chemical messenger. Without this receptor, the signal will have no effect. This is how the body ensures that only the correct target cells respond, preventing unnecessary or inappropriate activation. The signal pathway itself can be broken down into several key steps. The first messenger is a chemical signal, known as a ligand, that binds to a receptor protein on or within the target cell. Examples of ligands include hormones, neurotransmitters, or growth factors. Once the ligand binds to the receptor, this interaction activates the receptor and initiates the signaling process. The activated receptor triggers a cascade of events by activating one or more intracellular signal molecules. These molecules amplify and carry the signal inside the cell. Finally, the last signal molecule in the pathway modifies existing proteins or initiates the synthesis of new proteins. This leads to a specific cellular response, such as changes in gene expression, enzyme activation, or metabolic adjustments. This highly regulated process ensures that cells respond efficiently and appropriately to the signals they receive, playing a crucial role in processes such as growth, metabolism, and immune defense. This figure breaks down the 5 essential steps that most signal pathways follow, from the initial signal molecule to the final cellular response. Understanding these steps is critical to recognizing how cells process and respond to information. Step 1 begins with the signal molecule, often a hormone, neurotransmitter, or growth factor. This molecule travels through the extracellular space to reach its target cell. Next, the signal molecule binds to a membrane receptor protein. This binding event is highly specific and initiates a series of intracellular events by activating the receptor. Once the receptor is activated, it triggers the production or release of intracellular signal molecules. These molecules act as messengers, amplifying the signal and relaying it deeper into the cell. The intracellular signals then interact with and alter target proteins. These target proteins can be enzymes, structural proteins, or transcription factors. Finally, the altered target proteins generate the cellular response. This response could involve changes in enzyme activity, altered gene expression, or even structural changes within the cell, depending on the type of signal received. These 5 steps form a fundamental blueprint for signal transduction, ensuring that cells respond appropriately to their environment and maintain proper function within the body. Signal molecules can only influence target cells if they interact with the appropriate receptor, but where these receptors are located depends on the type of signal molecule involved. Lipophilic signal molecules, like steroid hormones, are fat-soluble and can easily diffuse through the lipid bilayer of the cell membrane. Once inside, they typically bind to cytosolic receptors found in the cell's cytoplasm or to nuclear receptors within the nucleus. By binding intracellularly, these signals often initiate changes in gene expression, leading to long-term effects like protein synthesis. In contrast, lipophobic signal molecules, such as polypeptide hormones, are water-soluble and cannot cross the hydrophobic core of the cell membrane. Instead, they rely on receptors located on the cell membrane. These receptors act as intermediaries, triggering intracellular signaling cascades when activated. This distinction between lipophilic and lipophobic signals is crucial because it determines how the signal is received and processed, ultimately affecting the speed and duration of the cellular response. Lipophilic signals often produce slower, long-lasting effects, while lipophobic signals typically lead to faster, short-term responses. We'll explore more about these mechanisms on the next slide. This figure highlights two key types of receptors: intracellular signal receptors and cell membrane receptors, and demonstrates how they respond differently to signal molecules. Let's start with Panel A, intracellular signal receptors. These receptors are located inside the cell, either in the cytoplasm or within the nucleus. Lipophilic signal molecules, like steroid hormones, can diffuse through the lipid bilayer of the cell membrane and bind to these internal receptors. This binding often triggers changes in gene expression, leading to slower but longer-lasting responses. For example, this type of signaling can regulate processes like growth, development, or metabolic changes that require sustained effects. On the other hand, in Panel B, cell membrane receptors are embedded in the cell membrane and interact with lipophobic signal molecules that cannot cross the membrane on their own. When a signal molecule binds to the receptor, it activates an intracellular signaling pathway that results in a rapid cellular response. These responses typically involve the activation of enzymes, ion channels, or secondary messengers, making them ideal for situations that require quick action, such as muscle contractions or hormone release. Together, these two types of receptors allow the body to regulate both immediate and long-term processes, maintaining flexibility and precision in cellular communication. There are four major types of membrane receptors. Receptor channels are open or close ion channels in response to ligand binding, crucial for rapid signaling in nerve and muscle cells. G-protein-coupled receptors activate intracellular pathways by triggering G proteins, often leading to second messenger production. Receptor-enzymes is where ligand binding activates enzymes, like tyrosine kinases, that initiate signaling cascades. Integrin receptors connect to the cytoskeleton, regulating cell shape, movement, and attachment. Membrane receptors are crucial for cells to detect and respond to external signals. There are four main categories of membrane receptors, each playing a specific role in signal transduction. Chemically gated ion channels, also known as ligand-gated ion channels or receptor channels, open or close in response to a chemical signal. This allows ions such as sodium, potassium, or calcium to flow into or out of the cell, rapidly changing the cell's electrical state and initiating a response. These are important in processes like nerve impulse transmission and muscle contraction. G-protein-coupled receptors, or GPCRs, are one of the most versatile and widespread types of receptors. When a ligand binds to a GPCR, it activates a G protein, which then triggers a cascade of intracellular events involving secondary messengers. GPCRs are involved in processes like sensory perception, hormone signaling, and immune responses. Receptor enzymes function by activating an intracellular enzyme upon ligand binding. Once activated, these enzymes catalyze reactions inside the cell, often leading to long-term changes, such as alterations in gene expression or cell growth. A well-known example is the receptor tyrosine kinase pathway. Integrin receptors are specialized for interactions between the cell and its extracellular environment. When activated, they connect the extracellular matrix to the cytoskeleton, helping regulate processes like cell adhesion, migration, and tissue repair. These categories of membrane receptors highlight the complexity and precision of cellular signaling, ensuring that the appropriate response is activated for each unique signal. Signal transduction is a critical process that allows cells to convert external signals into internal responses, ensuring effective communication and control of cellular activities. This process involves membrane proteins, which transmit information from one side of the membrane to the other. The key to signal transduction is the transducer, a protein that converts the signal from its original form into a form that the cell can use. Let's break down the sequence. The process begins with an extracellular signal, also known as the first messenger. This could be a hormone, neurotransmitter, or any other signaling molecule. When the first messenger binds to the receptor, it triggers the activation of protein kinases and amplifier enzymes inside the cell. Once activated, the intracellular signaling cascade begins with second messengers, which include molecules like cyclic AMP, calcium ions, or others. These messengers amplify the signal and carry it deeper into the cell. They can alter ion channel gating, increase intracellular calcium levels, or modify enzyme activity. For example, they may activate or deactivate enzymes like protein phosphatases, which are involved in regulating cellular responses. An important part of signal transduction is signal amplification. This occurs when amplifier enzymes increase the number of activated molecules, ensuring that a small external signal can produce a large internal response. Amplification is essential for processes that require quick and significant responses, such as muscle contraction or hormone release. This diagram illustrates the key steps of signal transduction, using the analogy of a radio to help explain how an external signal is detected, converted, and amplified within a system. It all begins with an external signal, which could be radio waves or, in biological terms, a hormone or neurotransmitter. This signal is detected by a receptor, either an antenna in the case of the radio or a membrane protein on the surface of a cell. Once the signal is detected, it is passed to a transducer, which converts the signal into a different form that the system can interpret. In the radio, the transducer converts radio waves into sound waves. Similarly, in the cell, the transducer converts the external signal into an intracellular message. To ensure that even a small signal can produce a significant response, an amplifier is activated. The amplifier increases the strength of the signal, just like how a radio amplifier boosts the sound output to fill a room with music. In cells, this amplification can involve secondary messengers, like cyclic AMP or calcium ions, to magnify the original signal. Finally, the system produces a response. For the radio, this response is the production of sound waves, while in a biological system, the response could be anything from enzyme activation to changes in gene expression. This example highlights the universal nature of signal transduction, emphasizing how biological systems, like radios, rely on precise detection, conversion, and amplification of signals to produce meaningful outcomes. This figure illustrates the key processes involved in biological signal transduction, breaking it down into basic and more complex pathways that convert external signals into specific cellular responses. On the left, for panel A, we see the basic signal transduction pathway. The process begins with a signal molecule, also known as the first messenger, binding to a membrane receptor protein. This activates the receptor and triggers the production of intracellular signal molecules, often referred to as second messengers. These messengers then activate or alter target proteins inside the cell, which ultimately lead to a specific cellular response, such as gene expression, enzyme activation, or ion channel regulation. On the right, for panel B, we see a more detailed view of signal transduction pathways. After the signal molecule binds to the membrane receptor, it initiates a cascade involving signal transduction proteins and amplifier enzymes. These enzymes produce large quantities of second messenger molecules, amplifying the original signal. Second messengers like calcium ions or cyclic AMP can activate various cellular pathways. For example, they may activate protein kinases, which phosphorylate target proteins, or they may increase intracellular calcium levels. Calcium can bind to proteins, such as calmodulin, to influence processes like muscle contraction or neurotransmitter release. In summary, these interconnected pathways ensure that even small external signals can be amplified and precisely regulated, allowing cells to respond appropriately to changes in their environment. This figure illustrates the GPCR adenylyl cyclase signaling pathway, a classic example of how G-protein-coupled receptors, or GPCRs, trigger signal transduction and amplify cellular responses. Let's follow the key steps. It all begins when a signal molecule, such as a hormone or neurotransmitter, binds to a G-protein-coupled receptor embedded in the cell membrane. This binding activates the G protein associated with the receptor. The activated G protein then turns on an enzyme called adenylyl cyclase, which serves as an amplifier enzyme. Adenylyl cyclase converts ATP, or adenosine triphosphate, into cyclic AMP, a powerful second messenger. This step amplifies the signal, as a single activated enzyme can produce many cyclic AMP molecules. The cyclic AMP molecules activate protein kinase A, or pKa, an enzyme that plays a crucial role in the pathway. Once activated, pKa phosphorylates specific target proteins, altering their activity and triggering a variety of cellular responses. These responses could involve changes in metabolism, gene expression, or even muscle contraction, depending on the type of signal received. The importance of this pathway lies in its ability to amplify the original signal, ensuring that even a small amount of external stimulus can result in a large and effective cellular response. By activating multiple downstream targets, the GPCR cyclic AMP pathway demonstrates how the body efficiently and rapidly responds to external stimuli. This table provides an overview of key second messenger pathways, showing how different molecules amplify external signals and trigger intracellular responses. Second messengers are essential for signal transduction cascades, as they allow small external signals to produce large, amplified cellular effects. Let's start with nucleotide-based second messengers. Cyclic AMP is derived from ATP and produced by adenylyl cyclase at the membrane. It is typically linked to GPCR pathways and activates protein kinase A, or pKa, leading to protein phosphorylation and alterations in ion channel activity. Cyclic GMP, on the other hand, is produced by guanylyl cyclase from GTP. It activates protein kinase G, or PKG, and is involved in processes like vasodilation and smooth muscle relaxation. Guanylyl cyclase can be membrane-bound or cytosolic and is often activated by nitric oxide. Next, we have lipid-derived second messengers. Inositol triphosphate is produced by the action of phospholipase C, which is linked to GPCR pathways. It releases calcium ions from intracellular stores, further amplifying the signal. DAG, or diacylglycerol, works alongside inositol triphosphate and activates protein kinase C, or PKC, which phosphorylates target proteins and regulates cellular responses like cell growth and differentiation. Finally, we have ions, particularly calcium, which serve as highly versatile second messengers. Released from internal stores or entering through ion channels, calcium binds to proteins like calmodulin, triggering changes in enzyme activity, muscle contraction, cytoskeletal movement, and more. In summary, this table highlights the diversity and specificity of second messengers. While they vary in their production and targets, they all play a role in amplifying signals and coordinating cellular responses with precision and efficiency. One of the fastest ways that cells generate a response is by altering the flow of ions through ligand-gated ion channels, also known as receptor channels. These channels are particularly important in excitable tissues, such as nerves and muscles, where rapid signaling is essential. The process begins when a ligand, such as a neurotransmitter, binds to a receptor channel protein on the cell membrane. This binding changes the channel's permeability, allowing specific ions to flow either influx into the cell or efflux out of the cell. The movement of ions, such as sodium, potassium, calcium, or chloride, can have immediate effects on the cell's electrical state. For example, an influx of positively charged ions, like sodium, can cause depolarization, a key step in the initiation of action potentials in nerve cells or muscle contractions. Because of their speed, ligand-gated ion channels are crucial for rapid responses in the body, such as reflexes, signal transmission along neurons, or the rhythmic contractions of the heart. Without this fast-acting pathway, many vital physiological functions would be slowed down, affecting overall bodily coordination and function. One of the most important and versatile intracellular messengers is calcium, a molecule we've already seen as a key player in signal transduction on previous slides. Calcium's role as a second messenger is critical because it can trigger a wide variety of cellular responses, from muscle contraction to neurotransmitter release. Calcium ions enter the cell through gated channels, which are tightly regulated to ensure precise control. These channels can be activated by voltage changes, as seen in excitable cells like neurons and muscle fibers, ligand binding, where a chemical signal opens the channel, or mechanical stimuli, such as stretch or pressure. Once inside the cell, calcium binds to proteins like calmodulin, which acts as a calcium sensor and regulator. When calcium binds to calmodulin, it activates various enzymes and pathways that lead to specific cellular effects. In summary, calcium's ability to act as a rapid, flexible, and precise messenger makes it essential for many life-sustaining processes, and understanding its regulation is key to understanding cellular communication. This figure highlights how calcium ions function as powerful intracellular messengers, playing a central role in a variety of cellular responses. Calcium signaling can be triggered by either an electrical signal or a chemical signal. Electrical signals open voltage-gated calcium ions channels on the cell membrane, allowing calcium to flow into the cell from the extracellular fluid. Chemical signals can trigger the release of calcium ions from internal stores, such as the endoplasmic reticulum, into the cytosol. As the concentration of calcium ions in the cytosol increases, it binds to specific proteins that act as calcium sensors, initiating different responses. Calcium binds to calmodulin, a key regulatory protein that activates various enzymes, altering protein activity and leading to changes in metabolism, gene expression, or other cellular processes. Calcium can also bind to other calcium-binding proteins, such as those involved in exocytosis, where vesicles fuse with the cell membrane to release neurotransmitters or hormones. Calcium's role in movement is particularly important in muscle contraction, where it interacts with actin and myosin filaments to generate force. In summary, calcium is a highly versatile second messenger, capable of triggering multiple pathways simultaneously. Its regulation is crucial for maintaining proper cellular function, as too much or too little calcium signaling can lead to serious disruptions in physiological processes. In addition to more traditional signaling molecules, soluble gases can act as short-acting paracrine or autocrine signals. These gases are unique because they diffuse quickly and have a short lifespan, making them ideal for fast, localized responses. The most well-known gaseous signal molecule is nitric oxide. Nitric oxide is produced by the endothelial cells of blood vessels and diffuses directly into the surrounding smooth muscle tissue. There, it triggers vasodilation, or the widening of blood vessels. This effect is critical for processes like regulating blood flow and controlling blood pressure. Nitric oxide's role in vasodilation has important clinical applications. For example, it's the mechanism behind drugs like nitroglycerin, which are used to treat conditions such as angina by increasing blood flow to the heart. Because gases like nitric oxide are short-lived, their effects are tightly regulated, preventing prolonged or unwanted signaling. This makes them highly effective for specific, rapid physiological responses. The arachidonic acid cascade is a key pathway in the production of signaling molecules known as eicosanoids, which play a major role in inflammation, pain, fever, and allergic responses. It all begins with membrane phospholipids. When triggered by a stimulus, an enzyme called phospholipase A2, or PLA2, converts these phospholipids into arachidonic acid within the cell's cytoplasm. Arachidonic acid can follow two main enzymatic pathways. The lipoxygenase pathway converts arachidonic acid into leukotrienes. These lipid-soluble paracrine molecules are primarily involved in allergic reactions and inflammation. For example, leukotrienes contribute to bronchoconstriction during asthma attacks and promote the recruitment of immune cells. The cyclooxygenase pathway, COX-1 and COX-2 enzymes, converts arachidonic acid into prostaglandins and thromboxanes. Prostaglandins regulate processes like pain, inflammation, and fever. They are also involved in the dilation of blood vessels during an inflammatory response. Thromboxanes play a key role in blood clotting by promoting platelet aggregation. Some of these eicosanoids also act as second messengers, amplifying the initial signal and ensuring that cellular responses are robust and localized. The importance of this cascade is reflected in the effectiveness of drugs like NSAIDs, or nonsteroidal anti-inflammatory drugs, which inhibit the COX enzymes to reduce pain, inflammation, and fever. By blocking key steps of the cascade, these drugs help manage symptoms of inflammation, while highlighting the significance of this pathway in maintaining homeostasis and responding to injury or infection. Signal pathways are highly regulated and can be modulated at various levels to ensure that cells respond appropriately to their environment. One of the key factors in signal modulation is the interaction between ligands and protein receptors. These receptors exhibit several important properties. Specificity ensures that a receptor will only bind to certain ligands, much like a lock and key. Saturation occurs when all available receptors are occupied, limiting the cellular response even if more ligand is present. Competition arises when multiple ligands compete for the same receptor. For example, norepinephrine and epinephrine can both bind to adrenergic receptors, influencing how the body responds to stress. We also need to distinguish between agonists and antagonists. An agonist is a ligand that activates the receptor and elicits the same response as the primary ligand. An antagonist binds to the receptor but blocks the primary ligand from binding, effectively preventing the intended response. An example is beta blockers, which block adrenergic receptors to lower blood pressure. Interestingly, one ligand may bind to multiple receptor types, and the response depends on the receptor involved. For instance, epinephrine can cause different effects in various tissues due to the specific receptor subtype it activates. Cells can also adjust their sensitivity through upregulation and downregulation. Downregulation involves decreasing the number of receptors, which reduces sensitivity to a signal. This can happen when a signal is too strong or sustained over time, helping prevent overstimulation. Upregulation is the opposite, where the cell increases the number of receptors to enhance sensitivity, often in response to low levels of a signal. Finally, many drugs target proteins involved in signal transduction pathways. For example, beta blockers reduce blood pressure by inhibiting the binding of epinephrine to adrenergic receptors, limiting the body's fight or flight response. This demonstrates how modulating signal pathways can have powerful therapeutic effects. This figure illustrates the roles of primary ligands, agonists, and antagonists in regulating receptor activity and cellular responses. On the left, we see the primary ligand, which is the natural or intended molecule that binds to the receptor and activates it. This activation triggers a cellular response, such as enzyme activation, ion channel opening, or gene expression. In the center, we have an agonist, which is a competing ligand that mimics the primary ligand. When an agonist binds to the receptor, it also activates the receptor and produces a similar response. Agonists are commonly used in medicine to enhance or mimic natural physiological effects. For example, albuterol, an agonist for beta-adrenergic receptors, is used to relax airway muscles in asthma treatment. On the right, we see the role of an antagonist, a ligand that binds to the receptor but blocks its activation. Unlike an agonist, an antagonist prevents the primary ligand from binding and does not trigger a response. For example, beta blockers are antagonists that block adrenergic receptors, reducing the effects of adrenaline and helping to lower blood pressure. This balance between agonists and antagonists highlights the complexity of signal regulation. By modulating receptor activity, the body and modern medicine can control critical processes like heart rate, muscle contraction, and hormone release, ensuring that physiological responses are appropriately fine-tuned. This figure demonstrates an important concept in physiology. The same signal molecule can produce different responses depending on the receptor subtype it binds to. In this example, the focus is on epinephrine and its effects on blood vessels. Epinephrine can bind to different isoforms of adrenergic receptors, including alpha receptors and beta-2 receptors. When epinephrine binds to an alpha receptor on an intestinal blood vessel, the vessel constricts. This is part of the body's fight or flight response, where blood flow is diverted away from the intestines to prioritize other organs like muscles and the brain. In contrast, when epinephrine binds to a beta-2 receptor on a skeletal muscle blood vessel, the vessel dilates. This increases blood flow to the muscles, providing them with oxygen and nutrients to support increased physical activity during a stressful event or exercise. This example highlights how the same chemical signal can have opposing effects based on the receptor type. This specificity ensures that the body can direct resources where they are most needed and fine-tune responses to changing conditions. This concept is essential in drug design. For example, drugs targeting beta-2 receptors are used to relax airways in asthma, while drugs targeting alpha receptors can help raise blood pressure when needed. By understanding receptor subtypes, treatments can be tailored to achieve the desired therapeutic effects. To maintain homeostasis, the body relies on long-distance signaling pathways, primarily involving the nervous and endocrine systems. These systems coordinate their responses using a response loop, which can be broken down into three key components. Input begins with a stimulus, such as a change in temperature, blood pressure, or glucose levels. The stimulus is detected by a sensor or receptor, which monitors the environment. Sensors have a threshold, meaning they only generate a signal when the stimulus reaches a certain intensity. Once detected, the input travels through an afferent pathway toward the next step in the loop. The input signal reaches an integrating center, typically in the brain or another regulatory organ. Here, the signal is compared to a set point, a reference value that represents the body's ideal conditions. If the stimulus deviates from the set point, the integrating center decides on an appropriate response. The decision made by the integrating center triggers an output signal, which travels through an efferent pathway to a target or effector organ. For example, the output signal may cause the heart to beat faster, muscles to contract, or glands to release hormones. This leads to a specific response that restores homeostasis. This feedback system ensures that the body can respond dynamically to changes and maintain internal stability, whether through immediate actions via the nervous system or longer-term regulation through hormones in the endocrine system. This diagram outlines the steps in a reflex pathway, which is an example of a response loop or feedback loop that helps maintain homeostasis in the body. Reflex pathways are commonly seen in both the nervous and endocrine systems, ensuring that the body can respond quickly and efficiently to internal or external changes. Let's break down the key steps. The process begins when a change occurs in the internal or external environment, such as a drop in temperature or an increase in blood pressure. Sensor. The stimulus is detected by a sensor or receptor, which monitors the variable and sends an input signal if the stimulus crosses a certain threshold. The input signal travels along an afferent pathway to an integrating center, which processes the information. The integrating center, often located in the brain or spinal cord, compares the incoming signal to a set point and decides whether a response is needed. If the variable has deviated too far from the set point, the integrating center generates an output signal. The output signal is sent through an efferent pathway to the target or effector organ. The target organ executes the appropriate response to correct the deviation. For example, the heart may beat faster to increase blood flow, or sweat glands may be activated to cool the body. This feedback loop ensures continuous monitoring and adjustment, helping to restore balance or maintain stable internal conditions. Reflex pathways can be negative feedback loops, where the response counteracts the stimulus, as seen in thermoregulation, or positive feedback loops, which amplify a response, as seen in childbirth contractions. The nervous and endocrine systems are the body's primary control systems, but they differ in how they regulate and respond to stimuli. Let's explore these differences across speed, duration of action, and how they code stimulus intensity. The nervous system is much faster than the endocrine system. Neural signals, transmitted through action potentials along neurons, can reach their targets in milliseconds. The endocrine system, on the other hand, relies on hormones traveling through the bloodstream, which takes more time to initiate a response. Neural responses are short-lived. Once the signal stops, the effect typically ceases almost immediately. Endocrine responses are longer-lasting because hormones can remain in circulation for extended periods, continuously influencing target cells. Both systems need a way to code the intensity of a stimulus, but they do so differently. The nervous system encodes intensity by increasing the frequency of action potentials. For example, a stronger stimulus will cause neurons to fire more rapidly. The endocrine system encodes intensity by adjusting the amount of hormone released. The higher the intensity of the stimulus, the greater the hormone concentration in the bloodstream. In summary, the nervous system excels at rapid, short-term responses, such as reflexes or muscle movements, while the endocrine system is better suited for long-term regulation, like growth, metabolism, and maintaining homeostasis. By working together, these systems allow the body to respond efficiently to both immediate and prolonged changes in its environment. Let's wrap up the key concepts covered in this chapter by summarizing the 5 main topics. We explored the various mechanisms that allow cells to communicate, including electrical signals, chemical signals, and the importance of local and long-distance communication in maintaining homeostasis. Signal transduction pathways were discussed in depth, showing how extracellular signals trigger intracellular responses. We examined the roles of first messengers, second messengers, and membrane receptors in these pathways. We highlighted important signaling molecules, such as calcium ions, nitric oxide, and lipid-derived molecules like eicosanoids. These molecules have diverse roles, ranging from triggering exocytosis to regulating pain and inflammation. We covered how cells fine-tune their responses through concepts like receptor specificity, saturation, competition, agonists, and antagonists. We also discussed how upregulation and downregulation enable dynamic changes in cell sensitivity. Finally, we examined the reflex pathway model, consisting of input, integration, and output stages. By comparing the nervous and endocrine systems, we learned how these control systems maintain stability in the body through feedback loops. Together, these topics provide a comprehensive understanding of how the body uses signaling mechanisms to adapt, respond, and maintain balance, forming the foundation for many physiological processes you'll encounter in later chapters.